know that you're here. Everybody on the board? Hey. All right. All right, a little bit of everything. First question. Hey. Thank you. Thank you. So 2/3 versus 1/3. Okay. So think about a bowling ball sitting at the top of a hill. Does it require energy to be input for it to roll downhill? No, it just rolls. What if I want to push that bowling ball uphill?
Yes.
Definitely requires energy. And we can think of a concentration gradient the same way, right? If something's going from a region of high concentration to low concentration, it doesn't require any additional energy, right? That's just simple diffusion. But if we're going to move something from a region of low concentration to high concentration, that's going to require an input of energy. So active transport, your clue here is active means we're spending energy. Active is always going to be moving against or up the concentration gradient. Should ask this one first. Remember, there are only two possible answers, A or B, for true or false. So I think a couple of people have chosen a possible answer that doesn't exist. All right. So hopefully we're mostly all right. Okay. All right, so diffusion shouldn't require an active input of energy. All right, if we're going from low to high, that's not diffusion. Remember, by definition, diffusion is going from high to low, which doesn't require any extra input of energy. Yeah, your hands are blocking. Same thing. There you go. Kathy, I think your hand's blocking it. No, you're just too far away. There you go. All right. All right. guys look things over. All right, so things might have to cross the plasma membrane, but they might not be able to. Remember we talked about glucose last time? Right, it's polar, so it's not going to cross the membrane effectively. So we're going to need something to help it cross, even if it's moving down its concentration gradient. Right, we're going to have to facilitate that process. Take a moment, talk to your neighbors. What's up? Convince them of your arguments, and then we'll reevaluate here.
Facilitated diffusion. It's not really mine. It's what's it called? Yeah. It's wrong. It could be simple diffusion. Yeah. I just think I hope it's the primary actor.
The last second I was like, wait.
Alright. So we shifted things a little bit. I would say about 50% of you changed your answer to the correct answer. And about 50% of us, not so much. So let's take a look at what we got. And then let's work through this. Okay, so we got primary active transport, simple diffusion, facilitated diffusion, and secondary active transport. How is it that we can eliminate, let's say, B right off the bat? It's using a protein, right? Simple diffusion is it just goes from high to low. We don't have to use anything else. Right, so we can eliminate B right away because we're using a protein in this process. Now, right, primary active transport, facilitated diffusion, and secondary active transport are going to use proteins. So the question is, can we eliminate any of those based on what we see? Primary and secondary both have the word active. So this requires an input of energy. Does this look like it requires an input of energy? Why not? Okay, so if you know primary active transport is using ATP, there's no ATP in this diagram, right? We're not spending any ATP here, so you should be able to eliminate that. But even if you didn't know that, how else would you know this shouldn't require an input of energy? Because it's going from a region of high concentration to a region of low concentration, right? So that's not something that's going to require any energy. So just based on that, I might not know what secondary active transport is yet, but I know it requires energy, and this doesn't look like it has to use any energy, so I can eliminate both of those. Okay, so that leaves us with facilitated. Let's see if I was right. All right, good.
Professor, the one before was facilitated too, right?
Correct. Only difference was one was a word problem, the other was an image. Do you want me to leave that up longer?
Yeah.
So, yeah, good. There you go. All right, you ready? All right, is everybody on the board? Not bad. All right, so let's go through the logic. Simple diffusion? Why not? Using proteins, okay. Is it active transport?
No.
No. Why not?
It's going from high to low.
Still going from high to low, so it doesn't take any additional input of energy. So by default, we're left with facilitated, right? This protein is helping diffusion through the membrane. Got it?
So what would like simple diffusion look like? Like literally just molecules going through?
Yeah. So what kind of molecules would just slip through the membrane? Small or tiny?
Yes.
But is it sufficient for them to be just tiny? They have to be uncharged or non-polar? Non-polar? Good. All right. So, what do you notice? What's different about this than the last one?
It's going through a couple of different proteins.
This is one protein, but it's showing you like stage one, a few seconds later, a few seconds after that. So what do you notice is happening with this protein compared to the last one? Yeah, this one's changing shape. I don't think I spelled diffusion right. There we go. Okay, so facilitated diffusion. We have different ways that we can do this. Do you remember what the last one, what the protein was labeled as? It was labeled as a channel protein. It's literally kind of like a pipe. But that pipe has a very specific shape, so only the molecules that have the right shape can go through there. But it's just like a channel. It's open all the time. Make sense? Okay. What's this one called?
Carrier protein?
A carrier protein. Right, this one is going to change conformation, which is a fancy way of saying it's changing shape. Right, so the concentration out here is higher, so when a glucose molecule diffuses into this region of the protein, it changes the shape. So right here it's kind of open facing this direction. But as it binds, it suddenly flips to being open, you do it, this direction. And so now the glucose molecule, it's not going that way, the only way it can go is out this direction. And so we're still going from high to low. We've got two different ways to do this so far.
So the glucose carrier protein has nothing to do with being active.
Correct, because we we didn't have to add any energy for this. We're still going from high to low. So this is still, right, facilitated diffusion. What do we call it if it doesn't require an input of energy? Passive. These are all forms of passive diffusion. So we had simple diffusion, right? So things that are small, uncharged, non-polar, can slip across the membrane. Yeah?
So the energy to open and close these channels is negligible, I guess?
Yeah, I mean, what it is the interaction of those two is going to cause a change in shape.
I see.
And whatever energy is necessary is just provided by they have a certain amount of kinetic energy as they interact.
Oh, okay.
So the channel protein refers to the opening in the actual protein barrier?
So a channel protein would just be like this. Right? And this guy just goes straight through. It's like a pipe. But, you know, we might have different shaped pipes. So, what's going to go through this one? Well, not this guy, but that one will. Right, so I can have different shapes for these proteins from all things go through, but it's going to just, you know, this one actually has to change shape in order for the solute to move across. I make these pictures as big as I possibly can, I swear. This was like the entire screen when I saved it, but it doesn't really apparently do much.
What's a yellow dot say?
The yellow dot. So we got red dots, which are solutes, and then we have a big green ball, which is a stimulus molecule or a ligand. I guess I can move my cursor out of the way, sorry. We're picking up on things. Right, this is just another example of facilitated diffusion. Now, how did you come to that conclusion? High to low. Okay, so it's a pretty good rule of thumb, right? You know if it's high to low, it's not using an extra input of energy. Right, and we don't see any ATP on here, so that's another good clue. Right, so what's different about this from the other ones that we just looked at? Let's dig it on this one. What is this thing? A ligand. What, you know what a ligand is? Ligand is just something that gets bound by something else, right? So this is a ligand for this protein. So do you remember when we were talking about non-competitive inhibitors in lab the other day?
Yeah.
Right? And we showed Pac-Man, and then, like, if you were in my class, Pac-Man sat on attack, and then his active site changed, right? Proteins are flexible. So if something binds somewhere else on the protein, that can change the shape of the protein in different places. So in this case, the binding of this ligand to the protein opens up the channel. So, right, these guys are just open all the time, right? But this is a gated channel protein. We can regulate whether or not this channel is open or closed. We leave it closed if there's no ligand around, but if we introduce the ligand, the protein changes shape, and now the solutes can diffuse down their concentration rate. Okay, so channel proteins, we can have non-gated, and we can have gated. And there's actually two different kinds. We can have stuff that's gated based on a ligand, but we can also have ones that change in response to voltage. Right? We can change the overall voltage on either side of the membrane. And when we're talking about voltage, what we mean is consider sort of the total number of positive and negative ions on either side of the membrane. Right? If they're not equal, then one side is going to be more positive and the other side is going to be more negative. And if we shift that, we can cause a protein to change shape. And so we'll see examples of this. That clock's totally not working, is it?
No.
It's now.
In the toilet.
If you can't read something you want me to clarify, let me know. Feel free to pick each other's brains as well.
Is that whole like yellow, it looks like a protein about the ATP or right?
The thing labeled ATP is ATP. The yellow is one of the solutes, right? That's potassium. The blue is sodium.
Okay.
Do you know
So the ATP is like
between
a
the
chemical.
primary, secondary?
No, ATP is just alcohol.
Because I know it's an active transport.
The black dots. Oh, yeah.
Let's see what we got.
I know.
I think, Alan, you gotta turn your card a little more vertical. There we go. Ever tilt your card down a little bit? There we go. All right. Okay, so this is a good sign, right? Nobody answered simple diffusion. Excellent, right? Almost nobody answered facilitated. Why is this not facilitated? We have a protein helping out, right?
It requires energy.
We're moving against the concentration gradients, right? You'll notice, right, here's the blue spheres, that's sodium. Which direction is sodium moving in this diagram? It's moving vertically. So, we're going from a region of low to high. So we've got to be using energy, right? And if I look at potassium, the squares, right? What's the potassium concentration out here? Low compared to high here. In which direction is potassium going? Potassium's coming this direction. So both sodium and potassium, even though they're moving in opposite directions, are moving up their concentration gradients. Right? So we know this has to be active. So the question is this primary active or secondary active? What do we say is the obvious distinguishing feature for primary active transport? We're spending ATP, right? The hydrolysis of ATP is directly going to power this process. So this is showing you snapshots, right? Like second one, a second later, a second after that, another second later, and then it would go back to this, okay? So initially, our sodium-potassium pump, by the way, if you see the term pump, pumping is not a passive process, right? That's active. Right, so our sodium-potassium pump is open facing the inside of the cell, right? It binds to a molecule of ATP and it binds to these 3 sodium ions, right? When it binds to ATP, it's going to hydrolyze it. So, when we hydrolyze ATP, we get a DP. What does the D stand for? Di for diphosphate. So we've lost the phosphate group. Where did that phosphate go? It's attached to the protein, right? Remember we said proteins can change shape, and one of the things that can influence protein shape is the covalent addition of different chemical groups. So here what we've done is we've covalently added that phosphate onto the protein. We've broken it off of ATP and stuck it onto the protein. There's a term for this. It's called phosphorylation, and you're going to see this a lot more later on. Phosphorylation just means you stuck a phosphate group on it, right? So we phosphorylated this protein when we hydrolyze ATP. That provides the energy to do what? What changed between here and here? Yeah, the shape of the protein changed. So now this side is closed off, this side has opened up, and the only direction our ions can move is to this side of the membrane. Okay, so then what's going to happen, so those ions move out, then what's going to happen is potassium can move in, and then we're actually going to hydrolyze that phosphate group off of the protein. So the protein can hydrolyze itself, that phosphate group comes off, and the shape goes back to this, right, open on the inside. So these ions move out of the channel into the cell. And then we can do it all over again. Spend another ATP, right? Pump stuff out, hydrolyze the phosphate group off, pump those ions in. Right, so the sodium-potassium pump is a classic example of primary active transport. The energy from hydrolyzing ATP directly provides the energy necessary to move solutes across the membrane. The bigger the word, the higher the concentration. I couldn't add brackets in this diagram. So this is good start. We've eliminated this. We've also eliminated this. Why? How do we know this is active transport?
ATP.
Okay, there's ATP being spent, so that's helpful. What else?
Going from low to high concentration.
Yeah, in both cases, no matter how we look at this, we're moving things, mostly moving things up their concentration gradient, right? So I've got a higher concentration of sodium outside than I have inside. So this is the sodium-potassium pump that we just looked at, right? Pumping sodium from low to high, right? And then potassium from low to high. So this is the same thing we just looked at in the other diagram. So if I was just looking at this, what kind of transport would this be? It would be primary, but we're considering this whole image, which makes it secondary active transport. So the question is, what makes it secondary? Well, in secondary active transport, we're going to use the energy from ATP indirectly. Okay, so let's look at this protein, right? This protein is what we call a symporter, which means it's moving more than one molecule in at a time, right? Or it could be moving them in opposite directions. It could be moving them in same direction. Symporter means they're moving in the same direction. So we can move more than one molecule at a time, depending on the type of protein we have. So, what's this thing? Glucose. So, is glucose moving up or down its concentration gradient? It's moving up its concentration gradient, right? Glucose, low concentration over here, high concentration on this side of the membrane.
Yes.
That's going to take energy, right? But is this protein hydrolyzing ATP? No. So where's the energy coming from to move glucose from low concentration to high concentration? Well, it's coming from the other solute, right? Our other solute is sodium. How is sodium moving? Sodium is diffusing down its concentration gradient, right? Sodium is high outside and low inside. So the energy of sodium diffusing down its concentration gradient is sufficient to provide the energy necessary for glucose to move up its concentration gradient. Does that make sense? So one thing is diffusing down its concentration gradient. When things are moving, what kind of energy is that?
Kinetic.
Kinetic. Okay, we'll talk about that more in a little bit. Right, but the diffusion down its concentration gradient supplies the energy necessary to move glucose up its concentration gradient. Well, where did the energy come from to make that concentration gradient, right? Anytime we see a concentration gradient, you think this is potential energy. But in order to make a concentration gradient, I had to spend energy at some point. So how did we make a sodium concentration gradient high to low? How did I get a high concentration of sodium out here and low in here?
Acid, the sodium potassium.
This guy, right? This guy is actively spending ATP to pump sodium out so that I have a high concentration of sodium outside and a low concentration inside. And then this protein says, oh, well, I can take advantage of that and just let sodium diffuse back into the cell and use the energy from that process to also bring in glucose. Right, so that is secondary active transport. The energy from the concentration gradient is powering the movement of glucose up its concentration gradient. But how did we get a concentration gradient? Well, we had to spend some energy in the form of ATP initially. So this is why we call it secondary, right? It's not the energy directly from ATP, it's indirectly, because we took the energy from ATP to build a concentration gradient. And then after that, we can use the energy from that concentration gradient to power the movement of another solute. That worksheet that I pointed you out to last time sort of walks you through these sorts of diagrams, ultimately, so that you can understand this one, right? This is the most challenging of those sorts of transport topics. So work your way through that one and then shoot me questions as you have them. Does anybody have any questions right away on transport? Yeah.
If it was just the sodium molecule,
would be facilitated, right? Because it would just be an ion going through a channel.
Okay, even though there's two of them.
Say that one more time. Maybe go back to your initial question.
So, on the left, the protein on the left, both glucose and sodium are going through.
Correct.
So, it would be the same thing as selective diffusion, because there's no energy.
If they were both moving down their concentration gradients?
Well, if glucose was going up, then sodium.
Oh, so if you're saying if glucose was diffusing this direction and sodium was diffusing this direction, so they're both going down their concentration gradients.
But like, the way it is now, would that be possible just by itself? Just the left protein?
No,
Okay,
because you wouldn't have a concentration gradient initially.
okay.
Yes. So, and you brought up another point, we can have symporters. So it's moving a solutes in the same direction. What if I told you something was an anti-porter? Yeah, then I'd be moving two solutes in opposite directions, right? So one would be going this way, one would be going this way. Symporter just means they're both going the same direction. So what would this be called? Symporter. What would this be? Yeah, technically I could call it an anti-porter because I'm pumping one thing this direction and one thing the other way. Awesome. Total side note, by the way, I started looking to your discussion online for your CRISPR stuff. It's good work. Keep it up. Remember, you've got, what is it, through Friday or Monday to get your pre-posted. But looks like you guys have some good conversation going. All right, so I think that's it for this. I think that was more beneficial than just flat out describing everything to you. Maybe, yes, no? Right, so we've got passive transport, simple diffusion or facilitated, active transport, primary active or secondary active transport. Right, and then facilitated diffusion, we said we have channel proteins which can be volted or ligand gated, and then we have carriers. Right, so you want to be familiar with sort of all of these different scenarios. Yes?
Can you give an example of voltage heated?
Yes, we'll come back to that. Actually, this would be a good point to work in neurons. I may have to, I don't know if I have a slide in here. I may have to pull one to give you a concrete example of that. Let me look and see what we got. If not, I'll bring this. All right. Another example of a channel protein that's super important are aquaporins. So aquaporins are, right, they're channels, but they are specifically going to let water molecules through. So if we kind of cut this thing in half and looked at it, right, what would you expect the interior of this to be like? Give me a property that you might expect if water is going to move through here. Hydrophilic. What about the surface over here? Hydrophobic, right? This line, this blue thing represents the membrane. So all the residues here that interact with the membrane should be hydrophobic. But the residues that are on the inside here are going to be hydrophilic. So I'm going to pass around a model of this guy. You can pop it in half. Right? You got the little water molecules going through. Gives you some idea of what these kind of things look like. Show you an animation here as well. Where is this guy? So it's going to highlight one water molecule here in yellow, just so you can kind of follow one as it travels through. There it goes. Now, this is not real time, because in real time, literally billions of water molecules will cross this per second. So yeah, it's a lot of movement. Now, water can cross the membrane, but not very effectively, right? Not very effectively at all. So, in order for cells to allow the movement of water across the membrane rapidly, they're going to use aquaporins. Okay, so we talked about ligand. talked about facilitated diffusion, carrier proteins. By the way, if we looked at concentration, let's say we're trying to move this from one side of the membrane to the other, and then we looked at the rate of diffusion into the cell, right? So as we increase the amount of glucose outside of the cell, what happens to the rate of diffusion? It goes up and up and up, and then it plateaus. Why does it plateau? Why am I not diffusing into the cell any faster?
You need a catalyst to make it jump up, right?
Not a catalyst, so
Or
this is an energy.
What do you call it? Something enzyme.
It doesn't have to be an enzyme.
No.
Eduardo? It's not quite equilibrium.
No more carrier protein, it's satisfied.
Yeah, there's no more proteins available. So think about it like a building with a bunch of doors, right? Only one person can go through a door at a time. If we have 10 doors to the building, how many people can go through like per second? 10, right? If I only have one person trying to get in the building, I get one person in per second. Okay, that's great. Now if I have 2 people, and I still have 10 doors, I can get 2 people per second. If I have 3 people, I can get 3 people per second. Eventually, I'm going to have enough people that all 10 doors are going to be used at the same time, right? It's not going to matter. I'm not going to get any faster than that. The most I could ever move is 10 people per second. Same idea here with trying to move molecules into your cell. All right, so we talked about the sodium-potassium pump, antiporters, ATP. We're going to come back to ATP. Sodium. All right, membrane potential. This is not something you often think about, but if we have something like the sodium-potassium pump, we are pumping ions in different directions. What we can create is a difference in the number of positive or negative ions on either side of a membrane, right? And so we can have, for example, far more positive ions outside than inside, right? So this is what we would call an electrochemical gradient, because I just have more protons positive outside than inside, right? I can have an electrochemical gradient. Why is it electrochemical? Well, I have a membrane potential where one side of the membrane is more negative than the other, and I have a concentration, right? So I have a higher concentration, which is low here. And then I could have the opposite, right? I could have a negative membrane potential here, but this particular ion is still going to have a higher concentration here versus here, right? So cells normally have a negative membrane potential, right? So something like this versus this. So the opposite sides have opposite charges. This is going to be really important when we get to, for example, cellular respiration, right? Because not only are we going to have things that want to diffuse down their concentration gradient, we also have a difference in electrical charge overall on either side of the membrane. So you have 2 forces working together to kind of move things from one side to the other. So there are proteins, right, electrogenic pumps, that are going to transport things to create a voltage. Sodium-potassium pump is one such example, right? That's the primary one. So we're pumping sodium one direction, potassium the other, right? We also have proton pumps, right? So we have proteins that specifically pump protons from one side of the membrane to the other. Proton pumps will be really important. In fact, do you guys remember the lysosome? What's unique about the interior environment of the lysosome? It's very acidic. How do I make an acidic environment? Increase the proton concentration. How could I do that? Yeah, use a protein in the membrane to pump protons into the lysosome to make an acidic environment, right? So that's exactly what they do. Endosomes and lysosomes have proton transporters. They'll spend energy, right? They'll spend ATP and pump protons from the cytoplasm into the environment inside the lysosome. So the pH here is going to go down, right? And that's actually really important. The proteins that function in here are all hydrolases. Hydrolases hydrolyze things. I don't want a protein that's supposed to be in here running around in my cell chewing up other proteins and carbohydrates and my DNA, right? That'd be a terrible thing. So one thing that your cell does is make sure that those proteins only function in a lower pH environment. That way, if for some reason they escaped and they're floating around in the cytoplasm, they're not going to work. Right? So we specifically send them to this compartment, and the environment in this compartment is a low pH, which is how those enzymes can function. All right, so take a second, talk with your neighbors, and see if you can classify what type or types of transport you see here.
I think it's secondary.
Right, the bottom one's secondary.
Yeah.
And then the first one, it looks like there's more hydrogen on side. So like, I think the hydrogen one is
Is it?
the proton pump. I think that one's secondary. And then
The energy? Is it in the overall? The secondary?
yeah, that's what I was getting. Oh, you meant all the parts secondary?
Right.
I think only the bottom. Oh, actually, no, wait, you're right. You think the top one's secondary? No, I think the bottom one's secondary because it has ATP. Oh, I see what you're saying now. I see what you're saying. Yeah, and then like secondary like changes it to ATP. Yeah, I don't know.
That makes sense, yeah.
Yeah,
The
that's-
primary on the bottom and the secondary on top. Overall.
Wait, what? I don't actually know. I think secondary will just have different elements to it, kind of the same way that this one does.
Alright, so who wants to explain what's going on? Volunteers?
Extra credit.
Nobody? I'll pick a name out of a hat. Find my. Where's my roster?
You get extra points.
No extra points. Alright, so let's see. I'm totally choosing at random. Who do I got? Maria. Maria, are you here? Alright. Maria. Okay. Okay.
And then there's a proton pump which is using ATP to put the hydrogen in the high so that it can activate.
So if we look at just the bottom, right, it's just primary active because we're spending ATP to move protons from low to high. If you look at the whole thing, this is secondary active because here we're using the energy from going from high to low to power low to high. Good. All right, so this is, for example, how plants are going to move sucrose into the vascular cells for transport throughout the rest of the tissues. Yeah.
Sorry, I'm pretty confused. In the other one, the glucose was going down instead of the sucrose. Does that have any indication? Like, does that point you into any direction whether it's
When you say down, what do you mean?
or like from going from concentration to concentration? But it's going the same direction as the hydrogen.
We could have a situation where they move in opposite directions. So that's a great question. Let's draw that scenario because I think that may help to clarify. Right? So let's say here's our membrane. And I have a high concentration here, low here. So I'm going to have some protein that's going to allow these guys to move this direction. Now, is this a sym-porter or an anti-porter? Sym-porter, because they're both moving in the same direction. But we could have an anti-porter, right? So if we were going to move something in the opposite direction, what would the concentration here be look like? Relative to this side. Should be
Hi.
low. Right, because this is moving down its concentration gradient. That's releasing the energy necessary to move this one up its concentration gradient. So you can't just look at which direction it's moving. In fact, it's really good that this diagram is sideways because when you say up or down, you don't want to be thinking vertically on the page or anything like that, right? You want to think about the concentration gradient. But the concentration gradients don't have to be the same on either side of the membrane we were just talking about. I'm not sure if that answered your question.
Yeah.
All right. So that's the bulk of transport, with the exception of bulk transport. How do cells move things in and out of the cell Costco style? How to move like a lot of material at once. Right here, we're just moving one thing at a time through a channel, or a couple of things at a time through a channel. I want to move a lot of material. Okay, yeah, I mean, you could add more proteins, but we're still, each protein's only moving a little bit at a time. Yeah, so we can do exo or endocytosis. Right? So this is what we talked about with the secretory system, where we made a vesicle chock full of a bunch of stuff that we need to secrete out of the cell. Or we can make a vesicle and pinch off the membrane to bring stuff into the cell. Whoa. So exocytosis is just releasing material outside of the cell. Endocytosis is bringing it in. Right? So pinocytosis is an example of just pinching off a piece of the membrane with the aqueous environment. So anything that's dissolved in the environment, you're just taking into the cell. Phagocytosis is sort of engulfing something. It's like cell eating. So for example, a white blood cell might engulf a bacteria cell to bring it in. Right? And then there are cases where we're going to bring in something based on what we call receptor-mediated endocytosis. So there's a protein that has to be recognized to trigger something to be brought into the cell. So our phospholipids, remember we said this is kind of like bubbles. I can fuse them together, I can pinch them apart. So here's exocytosis, right? Here's all the proteins, for example, that just got shipped to the plasma membrane. Where did this come from? Trans-Golgi, right? So it's being carried to this by the, what's carrying the transport vesicle? Motor protein, which one if we're going this direction towards the plasma membrane? Kinesin, and kinesin is walking along what? Microtubules. By the way, the exact question that I gave you was straight off the worksheet that you got. Right? Both that and the why are cells small question. So, hint, hint, wink, wink, nudge, nudge. If I give it to you, it's important. All right, so our membrane fuses, right? So these lipids actually become part of the plasma membrane now, right? It's not like the vesicle moves from this side to this side. We are literally fusing the membrane so that now the compartment here becomes continuous with the space outside of the cell. Okay, so this is bulk release. Yeah.
So does it like fuse with portions that don't have proteins in it?
So we're just not showing any proteins, but there can be
Oh.
proteins in the membrane here. There's gonna be proteins all along. Yeah. All right, so here's an example of this, right? Here's a vesicle with a bunch of, what is this, histamine? This one's histamine, right? So these are the cells that, when you have an allergic reaction, it's because you have cells that are chock full of histamine vesicles, and then when you get an allergic reaction, it's because you're triggering those cells to release all this histamine. This is why you get all the fun symptoms, and then you take an antihistamine in response. Right, so this vesicle is already sort of fused, and now this stuff is on its way outside of the cell. What kind of image is this? Like, what kind of microscope did we get this from? Electron microscope. How do we know that? Well, I can see the plasma membrane, which is really, really tiny, right? I'm looking at structures inside the cell, black and white, another good example, although sometimes they're false colored. Right? It's also what kind of, there's two types of microscopy that give us images for electron microscopy. You have scanning and transmission. Which one is this? Transmission. Transmission gives us like a 2D, kind of like a slice, right? Scanning, remember, gives us kind of that three-dimensional feel for things. Okay, so the opposite of this process, right, if we just took that diagram and flipped it around, it would look like this, right? There might be something outside the cell that we want to bring in. So the membrane is going to form a vesicle around whatever that is and bring it into the cell. And now the cell's got something it can work with. Oftentimes this gets sent to the endosome, which is like the lysosome. Kind of like starts out as an endosome and then becomes a lysosome. It's kind of interesting, the organelle changes. Don't have to know that. But that's often where this is going to go. But if that's where it's headed, what's going to happen to the thing inside of here? It's going to a lysosome. What's in the lysosome? Hydrolases, right? So whatever's in here is going to get hydrolyzed, broken down. So we're going to break down all the macromolecules that are in that compartment. So if I engulf the bacteria cell, right, I can destroy the bacteria cell when that reaches the lysosome. Right, so here's a picture of a white blood cell engulfing, I think this is a red blood cell, right? So the red blood cell's trying to get recycled. I don't know, maybe it had a malarial parasite living inside of it, right? Got recognized, it's got to be destroyed. So here's a white blood cell that's engulfing a bacteria. That bacteria is actually in the midst of cell division. You can see it's about to split into two cells, right? And its membrane is stretching around it, so we continue to do this, engulf it. Or this one's an amoeba that's going to eat this green algae cell, so it's like the blob trying to eat something. All right, so pinocytosis is the same thing, except that we're just taking in solute, basically. Right, so it's not like you're eating like a big physical object, it's just whatever's dissolved in the aqueous solution the cell's going to bring in. Okay, receptor-mediated endocytosis. This is when proteins on the surface are going to recognize whether or not they should pick up something from the exterior environment. Right, so if we have, so this little blue guy here, right, if it binds to the receptor, that will trigger this to form a vesicle. So you have these, what we call a clathrin-coated pit. So here's a bunch of clathrin and adapter molecules. The adapters look like Y shapes here. And then they're going to communicate with these guys here. And if this blue guy sticks to that protein changes shape, right? That shape changes the shape of these guys because they're all interacting. And then this thing starts to form and pull the membrane away. I'm going to show you an animation for this, because it makes it kind of clear. All right, so clathrin, this would be like one clathrin molecule. They form literally a cage. So the vesicle would be inside of this cage. So if we took a TEM, a transmission electron microscope, actually this might be TM, what is this? This is scanning. 3D, right? Along the membrane, you can see these clathrin pits, where the clathrin is sitting there waiting to get the final trigger and finish building that cage and pulling the membrane. So let me show you the animation for this. At least part of the animation.
What is the clathrin?
That's the protein. All right, maybe I'll show you this first and then we'll look at the 3D animation. Make it full screen. Right, so here's the membrane, here's the receptor protein, right, and the adapters. So if the cargo molecule binds to the receptor, then the adapter is going to change shape and now can grab onto clathrin. So, right, the clathrin is this green, funny shape here. So now it will start to stick together to make this sort of cage-like structure. You notice it's kind of pulling the membrane as it forms this sphere. And then you have some proteins that will sort of pinch this off so that we end up pulling in the vesicle, but the vesicle is coated with clathrin. That's how we end up making a vesicle. And then these guys will disassemble, and now we have brought this into the cell, and then this can go to the lysosome or the endosome. But we only formed this vesicle because this receptor bound to whatever this molecule was. All right. So here's the receptor. On the surface, this is looking sort of at the surface from inside the cell. So that's an adapter protein. All right, so right, the adapter is going to bind to clathrin. So these are clathrin molecules. And the way they assemble, they start to make this cage. Right, so see how the membrane is kind of being pulled in? Right, so these purple things that you just saw sort of through the translucent membrane are what's being pulled in because they're binding to the receptors.
How are we going so fast?
That's just, this is room temperature, body temperature, right? This is just, I mean, this isn't even necessarily the speed scale. Whoa. So you pinch off the membrane, and now you've got the cage enveloping the vesicle. So we've successfully brought it into the cell. And then you have proteins that will help that cage disassemble now that we've brought it in. By the way, why are all these proteins interacting with each other? That's how they're doing it. Why are they able to do it? Why would two proteins interact with each other?
They recognize each other.
Why do they recognize each other? They have the right shape and chemistry. All right. I don't know what that is. Okay, so that's the end of transport. A little slower than I was hoping. So hopefully you guys have a good handle on transport. Ask lots of questions. The other thing I wanted to note is I saw maybe 4 of you in my office hours before exam 1. I should see all of you. My office hours, I mean, I love that time because I can get a lot of stuff done. But that's not why I'm paid to be there, right? My office hours, it doesn't mean like that's my office time and leave me alone. That means it's my office time and you should come bug me with every question you can possibly think of. So if you can make it to office hours, I highly encourage you not to wait to see if I can answer your 3-2-1 online, but to come ask questions during those time periods. And if you can't, right, schedule a time to meet with me. In fact, one of the things I'll do is I'm gonna, for the exam, give you guys a reflection exercise to think back about, for example, how you thought the exam went, how did you prepare for it, what would you change, did you come to office hours, if not, why didn't you, right? And if you scored at a certain level, I'm going to say you are required to come see me. And if you scored above a certain level, I'm gonna tell you you're required to come see me. Okay, good, right? So, you know, honestly, I TA'd for 7 years, long before I ever taught. And one of the most important things I learned is that the one-on-one time with an instructor is some of the most valuable time you can have. So when I set that time aside, it's kind of wasted time if it's not spent with you guys. Right? So I just want to emphasize that as much as I possibly can. Ready for energy? Okay, so we're going to prime ourselves with energy. You might need a primer of energy. It's been a long day. So first of all, what is energy? Like, let's just, you know, I drink a Red Bull, drink my coffee, drink my Dr. Pepper. Is that really energy? What is energy? Capacity to do work. Okay, now if you've taken a physics class, right, work has a very specific definition. Right, but another way to think of this is just we need to transform material, right, matter. We need to transform matter in some way, right? That can be making or breaking chemical bonds. It can be literally moving things from one place to another, right? Those are all forms of different type of work, and that takes energy. So energy is what allows that to take place. So what are the two basic forms? We've talked about this so far a couple of times. All right, we've got potential and kinetic. So potential is stored energy due to location or structure. So my pointer has a higher amount of potential energy here than it does here because of position. Right? When I look at the structure of a molecule, there is potential energy in the chemical bonds because of the bond and where those electrons are located. Right? But this is energy that hasn't been released. So forms of this, gravitational potential, right? So like here versus here. Chemical bonds. Chemical gradients. And electrochemical gradients. So both of those we've seen so far. All right, now, gravitational potential, to the best of my knowledge, is not something that biological systems take advantage of at a cellular level. Now, I guess we can have all kinds of fun with that when we talk about like jumping out of an airplane. But that's entirely different, right? So cells are going to utilize chemical bonds and gradients. Right, so kinetic energy is energy in motion, i.e. energy that has been released. So what are some forms of kinetic energy you can think of? What? Okay, so something in motion like a ball, something moving, dropping. What else?
Molecules bouncing around.
Okay, so that would be analogous to molecules bouncing around. What else?
Thermal.
That's so that would be thermal, right? Like the energy that molecules have.
When you break a bomb?
Okay, light. So that's an example. Good. Somebody else said something? All right. So thermal energy is usually the first one we think of. Right? Molecules jiggling around. They have a certain amount of motion. So, by the way, heat energy is energy, thermal energy that's being transferred. Right? So if I have like a cube of ice and I put it on my desktop, right, that energy from the desk is going to transfer from the desk into the ice cube. Like that's what we mean when we're talking about heat. We're talking about transferring. An electric current. This is the flow of charged particles. Radiation. What we're thinking is like, for example, photons. I guess other types of radiation, right? Like if you've got emitting alpha, beta particles, that's also a form of kinetic energy, right? This is going to be really important later on, right? And then mechanical energy, i.e. structural movement. Okay, these are all forms of kinetic energy. The breaking of covalent bonds, or the flow of solutes from high to low concentration, those are forms of kinetic energy that's being released. And again, some of these are ones that biological systems really rely on. So we're going to see the flow of charged particles later on. We're going to see light. What kind of process would this be used in, by the way?
Photosynthesis.
Photosynthesis, right? This actually powers photosynthesis. Moving things around the cells, right? Mechanical energy. We're going to look at how chemical gradients and the flow of things down their concentration gradients is actually essential for powering cells, right? And then we're going to look at how breaking covalent bonds is important as well. Right, so these are all different forms of kinetic energy. Okay. So, how many of you guys are familiar with the laws of thermodynamics? How many of you guys aren't sure? Like maybe I've heard about it, maybe not. All right. There's two laws of thermodynamics. And I don't know about you, but when we started talking about thermodynamics in biology class, I was like, why on earth do I care? Right? The first reason I'll tell you is because these are sort of laws of the universe, right? So biological systems follow these laws, right? So we're going to understand how biological systems function if we understand the first and second law of thermodynamics, right? The second reason is it explains why I have to eat every day. All right, so let's see if we can think about why this is. Organisms are open systems, right? What that means is that you have a transfer of energy between the system and the surroundings all the time. Right? So biological entities are transferring energy between themselves and the system all the time. Now, the first law of thermodynamics, energy cannot be created or destroyed. Okay, so I'm going to get really particular, and I'm going to expect you to be very particular about me. Sometimes I use anthropomorphic language a lot, right? Which means I'm sort of giving human attributes to things that aren't human at all. So I'll say like the enzyme wants to do this, or I'm going to kill an enzyme, right? Those are analogies. They're not really true things, right? So be really careful when you're talking about energy. We cannot make or destroy energy. The only thing we can do is transform it from one form into another. So if we say, oh, the mitochondria is the powerhouse of the cell, it makes energy. Call me out if I say that, right?
Okay.
Like, you have full permission to be like, right? You were particular on you guys, like I'm particular on you guys, you should be equally particular on me. So make sure that if I say something that sounds funky like that, you call me out. We can only transform from one form into another. So if I start off with this much energy, and I go through some process, I'm going to have the same amount of energy later on. It may not be all in the same form, but I'll still have the same amount of total energy. Does that make sense?
Right.
So can't come up with any extra. I'm not going to lose anything, like it's not going to disappear. It's just going to be transformed into different forms. All right. The second law of thermodynamics is essentially telling us what happens when we do transform energy from one form into another. Right? So if I start out with this much energy, and we'll call this useful energy, when I transform this energy, right, some of it's still going to be useful, and we're going to call this free energy. We'll come back to what that means later. But some of that energy is going to be unusable. It's not going to be useful energy. So if I started off with, we'll call this 100%, we go through some transformation, right? Maybe 80% of it is still useful and 20% of it's not useful. It's still there, but it's not in a form I can use. Any idea what this form typically is? Heat, right? most of the time, we are going to lose useful energy because we've transformed it into heat energy. And for biological systems, heat energy cannot be used to do work. So it's not useful for a biological system. That's not to say it's not important, right? If I'm freezing to death, my body is going to start converting energy and producing a lot of heat energy. And that's useful to me, right? Hopefully it keeps me alive. But it's not doing any actual cellular work. Right? So every time we do an energy transformation, more and more of that energy
Is unusable.
is unusable. So I eat a hamburger, and then 6 hours later I gotta eat another hamburger. Why?
Could you use it?
Because as I transformed the energy from that hamburger to do things in my cells, less and less of that energy was useful. Eventually, all that energy is transformed into heat energy. So I gotta eat another hamburger. I gotta eat again. Right? So now you know one reason why you have to eat. By the way, what's the other reason you have to eat?
Does it taste good?
It tastes good.
Wouldn't you be like nourishing your body?
Oh, because we can't produce all the stuff.
Nourishing, what do you mean by that?
We can't produce all the stuff we need to keep ourselves running. Like amino acids.
Okay, so there are some things we have to get from our diet because we can't make it. But if I have to make something, where am I getting the building blocks from?
Proteins.
Where am I getting some proteins?
Yeah. I mean, from the ribosomes.
Your food.
Ribosomes are going to make it, but where am I getting the amino acids then?
From meat.
Yeah, from the hamburger I ate, right? Like the hamburger patty is just a bunch of proteins. So if I eat the hamburger patty, I break the proteins down into amino acids, and then my body takes those amino acids and says, oh, I actually need to build this protein. So you eat for two reasons. One is to provide energy to your body to do cellular work, all the things that you have to do to be alive. And the other is to provide the building blocks that you need to build all the molecules in yourself. Right? So I don't remember if I used this analogy with you, but it's like somebody built a very nice Lego house. I don't want a Lego house. I want a Lego truck. So what am I going to do? I'm going to tear the house down. I have all the building blocks I need. Now I build my Lego truck, right? I need protein X. So what do? I eat a hamburger. I break down those proteins into the amino acids, the building blocks. I take those amino acids and now I can build protein X. Okay, so that's why you eat, right? One of those reasons is I need building blocks, and the second is the second law of thermodynamics means I'm always going to need to bring in energy because that energy is going to be lost as heat. All right. How are you doing? It's time? I think this is a good point for us to call it. So, I want you to make sure that you are familiar with the first and second laws for when you come back on Thursday. And then we're going to get through enzymes and energy on Thursday.
That is the wrong decision to make.